everyone, and welcome to Make Better Decisions for Cell Therapy Manufacturing, leveraging image-enhanced flow cytometry for predictive donor material characterization. I'm Matt Thorne of Labroots, and I'll be your moderator for today's event. Today's educational webinar is presented by Labroots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You can also submit any technical issues there as well if you're having trouble seeing or hearing, and we'll address those directly. I'd like to now welcome our speakers, Nicole Madfis, Global Product Manager for Thermo Fisher Scientific, and Jasmine Padilla, R&D Scientist, Cell Therapy Innovations, Thermo Fisher Scientific. For a complete biography on our speakers, click on the biography window on the left of your screen. Nicole and Jasmine, you can now begin your presentations.
Thank you for the introduction. Today, we want to present to you one of the exciting projects we're developing for cell and gene therapy workflow using imaging-enabled flow cytometry. Today's presentation will be given by myself and my colleague, Jasmine. She will present work performed by her and the bioinformatics team regarding the creation of a predictive AI model that can be used to evaluate donor material for cellular therapeutics. In our agenda, we will discuss the benefits of imaging-enabled flow cytometry and where we see applications in the cell therapy workflow using the Attune CytPix. Jasmine will then discuss her results. And then I will talk about how morphology analysis on the CytPix can help to improve the accuracy of flow cytometry data. What is image-enhanced flow cytometry? It is time to get excited about something new. Traditional flow cytometry provides the visual representation of your cells on a scatter plot in the same way, as dots in a relative position to one another. This is regardless of various complexities within subpopulations, cell lineages, and between dead or healthy cells. We trust that the dot is a cell and go through various downstream processes such as sorting, genomics, and microscopy to verify. It's time to move away from the faith-based approach to flow. With imaging-enhanced flow cytometry, there's less guessing what your detector results mean. Even slight changes to reagent titration, isolation protocols, or instrument settings can change your results. It's fundamental to confirm what you're analyzing. With imaging, you have visual evidence and the ability to quantify what you're seeing, providing a whole new data set. This can help to enhance the statistical power of already sensitive flow cytometry data and expand morphological analysis beyond traditional flow and microscopy. The combination of high-speed image capture and machine learning make this technology a reality and easy to use. On the Attune CytPix, you can capture real bright field images, and with the assistance of machine learning, analyze those images at scale. And if prompted, to a depth that is difficult to discern with the human eye. You can clearly inspect objects within the images, confirm that the machine learning models are counting them correctly, and determine whether they are beads or cells or something else. In this example, gating around single particles or doublets is made easy. We provided a method to consider morphology of every cell as an additional set of data that can be integrated with traditional flow data. This enables improved population resolution without the use of labels. Over to you, Jasmine.
Thank you, Nicole. Now I'll go into leveraging image-enhanced flow cytometry for advanced cell therapy insights. Flow cytometry is a cornerstone of cell therapy manufacturing workflows, providing critical insights into cell processing and cell state through the manufacturing process. Here's a broad overview of our closed modular cell therapy manufacturing process and how flow cytometry plays a role through the entirety of it. The workflow begins with cell isolation, activation, and bead removal using our magnetic bead-based technology facilitated by the Gibco CTS Dynaselect magnetic separation system. Next, cells can be washed, concentrated, and buffer exchanged into the appropriate media using the Gibco CTS Rotea counterflow centrifugation system. For large-scale gene editing, our Gibco CTS Xenon electroporation system enables efficient electroporation. Following gene editing, CAR T-cell expansion is carried out using GMP-compliant bioprocessing equipment such as the Thermo Fisher Scientific DynaDrive single-use bioreactors. Once the expansion phase is complete, the cells can be washed, filled, finished, and cryopreserved using the cryomed-controlled rate freezer. At the final quality control step, the end product cells undergo comprehensive analysis, including genomic, proteomic, and cellular assessments, as well as identity, purity, and sterility testing to ensure they are safe and functional for the patient. The Invitrogen Attune CytPix flow cytometer plays a key role not only in this final QC stage, but also throughout earlier workflow stages. As mentioned in the earlier slide, the CytPix flow cytometer plays a key role through the entirety of the end-to-end cell therapy workflow. At the start of the process, during cell isolation and activation, it is used to immunophenotype the donor material, establish a baseline receptor profile, and evaluate key process metrics such as target cell yield, purity, and isolation efficiency. During cell engineering, flow cytometry provides critical insights into cell population and genome editing success. It also enables the monitoring and quantification of cell activation and exhaustion marker expression post-editing. Throughout cell expansion, flow cytometry is instrumental in tracking the dynamics of various T cell subgroups, as well as observing shifts in activation and exhaustion markers. Finally, at the end of expansion, it is used once again to assess the purity and perform quality control characterization of the final cell product, ensuring the highest quality for downstream applications. These workflows stand to gain even greater efficiencies and deeper insights through the integration of imaging flow cytometry, which offers enhanced analytical capabilities. While traditional flow cytometry offers robust capabilities in cell therapy workflows, imaging flow cytometry adds a new dimension of analysis. By combining the power of flow cytometry with high-resolution imaging, it enables more detailed characterization of cell morphology, unlocking deeper insights into cell state and function throughout manufacturing. The Invitrogen Attune CytPix flow cytometer exemplifies this innovation. This advanced cell analyzer utilizes a high-speed bright field camera to capture images of individual events as they pass through the flow cell. By pairing morphology data from the CytPix camera with fluorescent marker staining, we gain a more comprehensive understanding of the cells involved in cell therapy manufacturing workflows. With imaging flow cytometry, we gain another data set across the process. At the start of the workflow, during the cell isolation stage, morphology data provides valuable insights into the quality and health of the starting donor material, allowing you to visually inspect key aspects of cell morphology, such as size and shape. It can also aid in validating your gating strategy directly on the instrument, allowing you to visually inspect and assess whether you have, for example, all singlets and live cells captured. This capability extends across other stages of the workflow, including cell engineering, cell expansion, end of expansion, and lot release quality control and characterization, offering consistent and actionable insights throughout the process. Now I will talk about the results of a project that I've been working on, which involves predictive donor T-cell profiling using morphology and flow markers. Before we dive into how morphology data can be utilized, it's important to first define the various aspects of cell morphology. These can be characterized as follows. First, there is size. This includes measurements such as perimeter, area, and diameter, which provide a fundamental understanding of cell dimensions. There is shape. This refers to attributes like circularity or ellipticity, describing the overall form of the cell. There's cell texture. Metrics such as kurtosis, skewness, and coefficient of variation of intensity help characterize the cell surface or internal granularity. There's color or intensity. This includes the degree of shading from white to black and measures like maximum, minimum, and average intensity, which provide insights into structural features. Then there's aggregation. This indicates whether the sample contains single cells or clusters, helping differentiate between individual events and aggregated populations. By breaking down cell morphology into these distinct components, we can more effectively leverage this information for analysis. Using the morphology and marker expression data obtained with the Invitrogen Attune CytPix flow cytometer, we began developing a model to screen and characterize starting donor material in our CAR-T workflow. To start, we sought to better understand cell status and proliferation in unmodified T-cell expansion first. This proof of concept serves as a stepping stone for implementing image-based predictive models into CAR-T workflows. Ultimately, our goal is to build a model capable of predicting overall cell performance, expansion potential, and health from the outset of the workflow. For the proof of concept project, we collected flow marker expression and morphology data from unmodified T-cell expansions across multiple donors. The CytPix software allows us to retrieve the ACS file from our experiment plate, which contains both marker and morphology data, once the samples have been run and images processed. From our gating strategy, we annotated specific cell populations, creating a comprehensive cell database. This database includes single-cell data that encapsulates morphology, annotations, and flow marker intensities, forming the foundation of our project. We have so far collected 7 full expansion datasets on unmodified T cells and split them into 2 subgroups. One subgroup that gets only morphology-based analysis, and another that gets a morphology and marker-based analysis. For each subgroup, we rank the donors based on their relative ability to expand through day 9. For each donor, we also collected donor biometrics such as age, BMI, gender, and ethnicity so that we can keep an eye out for any correlations to any of these metrics as we build our model. However, thus far, there does not seem to be any correlation between expansion capability and these collected biometrics. For the morphology-based analysis, we used unsupervised learning without fluorescent marker staining to identify cell populations in the day 0 peripheral blood mononuclear cell samples for 3 donors to see if it reveals morphology profiles of these samples that could correlate to expansion potential. On the left, we have a projection of population morphology in a 2D space. We see a clear cluster between the donor cells that showed best expansion, aka donor 3 in the red, and the donor cells that showed worse expansion in the teal and orange, aka donor 1 and 4 respectively. On the right, we see a more thorough pinpointing of different morphology profiles. The clustering reveals that donor 3, the donor that expanded the best, has a consistent population morphology, whereas donor 1 and 4 have a very diverse and heterogeneous morphology population on day 0. We then pulled some images that belong to each of the different morphology clusters, and we can see that donor 3, which was our best performer, has very smooth and circular cells, and this was pretty consistent across all of the day 0 samples from this donor. On the other hand, Donor 1 and 4 had many different profiles that varied in shape and texture in their own unique way. This initial exploration of whether morphology parameters can correlate to expansion capacity revealed that certain morphology parameters such as smoothness and circularity, as well as the heterogeneity of cell morphologies in the day zero population, can potentially offer information about how the cell may behave in the future. The morphology-only analysis gave us a general description of the cell populations on day zero according to morphology parameters only. We added another dataset to our analysis, which is the marker expression based on fluorescent antibody staining and detection. This time, we did clustering using both datasets to pinpoint cell subpopulations in day zero PBMCs across 4 donors and tried to see any correlation between day zero data and T cell fold expansion at late phase of expansion. On the left is a projection of the clustering based on flow markers and morphology metrics, and there were 19 identified unique clusters. When we project the donor samples to the cluster space, we see that the donors that expanded best, which is donor 6 in the teal and donor 9 in the yellow, clustered together, and the donors that expanded worse, which is donor 2 in the red and donor 7 in the orange, tended to also cluster together. Additionally, when we look at the enrichment across the clusters, on the right, we see that the best expanding donors, donor 6 and 9, shows very different enrichment patterns than the worst expanding donors, donor 2 and 7, which is a strong indication for differentiation between donors with better versus worse expansion. From the 19 communities identified, we overlaid the cell annotations based on what we stained the cells with and what they express to show us the identity of the cells, CD19, 56, 3, or 14, in each of these clusters. We see that a majority of the cells are CD3 positive, CD28 positive cells, which makes sense as the sample was collected post-isolation. However, it reveals that amongst the CD3 positive, CD28 positive population, there are very distinct morphology profiles that exist, and that the donors that expanded better tended to cluster together within these 19 communities. Again, we see that the heterogeneity of cell morphologies in the day zero population can offer information about how the cell may behave in the future. To better understand what morphology parameters were most significant, we also looked at the enrichment across morphology metrics between the 4 donors and saw that there were very distinct differential patterns in the better versus worse expanding donors. Of note, parameters related to texture, brightness, and distribution of signal intensity, such as skewness, kurtosis, entropy, and average norm intensity, and parameters related to shape, such as eccentricity, area, circularity, and diameter, were largely what significantly distinguished between the better versus worse performing donors at day zero. These initial exploration experiments confirmed to us the value of looking into morphology to give deeper insight into donor variability and how it may play an effect in performance in the future. As we continue to add more datasets, we aim to continue learning and growing our understanding of the biological relevance of the morphology images and the parameters captured by the Attune CytPix. The goal for this project is to see if we could build a predictive model to better characterize donor material from the beginning to enable smart manufacturing and process development decisions early on before extensive time, resources, and labor are put in. This capability helps us to uncover innate variability and the unique cellular responses to different steps of the process, which often vary from donor to donor. Such insights enable the development of workflows tailored to these differences, ultimately enhancing efficiency and effectiveness of the cellular product. These advancements have the potential to transform cell therapy manufacturing by reducing costs, time, and resources required to optimize workflows. As a result, manufacturing bottlenecks can be alleviated, increasing accessibility for patients and accelerating the delivery of life-saving therapies. This could also be largely beneficial to the allogeneic space for both CAR-T and CAR-NK therapies, where healthy donor cells are processed to generate several doses down the line. If we can better understand donor variability through more advanced characterization from the beginning, we can then better understand ways we can tune the process in a smart manner ahead of time to ensure a more robust and efficacious end product. I'll pass it off to you, Nicole.
Thank you. Imaging on an acoustically focused flow cytometer. The Attune CytPix is a benchtop instrument and the world's only acoustic focusing flow cytometer with imaging capabilities. As part of the Attune family, it is designed for cell therapy workflows with flexible and fast run rates up to 1,000 microliters per minute. It is clog resistant and comes in 4 laser, blue, red, violet, and yellow configurations with 6-channel violet laser options that can be used for discerning red blood cells and whole blood samples. It simply excels at taking high-quality, bright field images up to 6,000 images per second with a visual detection down to 0.8 micron. This is a sampling of the many events we have imaged on the CytPix. Acoustic focusing and positive displacement technologies allows users to obtain high-resolution data, including images, while reducing clogs, so that we're able to image events that we wouldn't dare put through most instruments. With this in mind, the CytPix is amenable to many applications. If your flow cytometer doesn't have acoustic focusing, then you're introducing sources of error in your data. It allows users to have focused images at high flow rates while keeping a consistent plane of focus. Whether it's FCS data or high quality images, we are able to achieve accurate and robust data. One of the hurdles to surpass in imaging flow cytometry is positioning the particles in the flow stream that is consistent from event to event, especially as you increase the flow rate. This is critical as even a small position difference can cause the cells to fall in and out of focus, as you see here. This diagram shows a hydrodynamically focused flow that is more than 10 microns in diameter and less than 6 micron depth of field. So in this sense, it's very easy for events with hydrodynamic focusing alone to drift in and out of focus. And you can see this on the top middle. With traditional hydrodynamic focusing, not all particles flow through the focus position. They are bouncing around in the field of view. By adding acoustic focusing, the CytPix can help to provide consistent positioning of a cell or events in 3 dimensions. Versatility and label-free morphology analysis. We don't expect users to visually inspect thousands of images, so we created machine learning models that could be taught to analyze immune cells. We trained the models with various workflow conditions. It was a matrix of leukocytes, beads, treatments. These were evaluated for model performance at different camera settings, injection rates, and with cells exposed to different buffers. This is what we refer to as pre-trained learning models. Our software provides automated image analysis and is applicable for most flow cytometry workflows. It combines 2 workflows into one. It assists users to make decisions based on data and not user judgment. So lack of reproducibility brought in by user bias and gating mistakes are reduced. They can also process images at a high speed up to 1,000 images per second through high throughput sample analysis, and is also 21 CFR compliant. Below, you can see a diagram of the workflow. It is performed entirely on the Attune platform without having to export the data. Users can acquire the data, choose desired sample, gates, and models for image processing in order to control the data footprint. And then they can analyze morphology as parameters. These morphology parameters can be combined with or without the fluorescence and light scatter parameters. Users can confirm the accuracy of the model by examining the borders of the image processing mask. You can see this here in green in the images. This is the outline around the cell that you see. This is a quality control measure and provides confidence to the user. The image processing models allows the identification of objects in the foreground of an image and measure specific patterns and characteristics within the image, and then transforming the image data to digital data. The digital data are characterized as morphology parameters, and we've divided these into 5 feature categories. These features can help to discern cell responses such as blebbing, swelling, or shrinkage. These morphology parameters are label-free, correlating to various shapes and intensity features, such as Jasmine explained earlier. Some examples include being able to measure how circular a cell is. That can convey the health status of a cell. When cells are healthy, they tend to be more round compared to a shriveled or pocked, less circular appearance when they start to die. Surface topology in bright field images is conferred through light and dark spots on the surface of the cell. One of the parameters is called entropy intensity. If you recall entropy from your high school chemistry class, it can be referred to as a measure of complexity. So a cell with high entropy may appear in images to be rough, and they could be dying, stressed, or highly differentiated, and that is in comparison to other cell types in the sample. How morphology assessment improves flow data. Accurate singlet gating is one of the most problematic issues, as some dots will fall outside of this typical polygon gating scheme, and we may consider larger proportion of these to be singlets. And as you can see here below in these diagrams, different gating schemes are used to analyze single cells. However, this is with little verification. The object features are used for counting objects. With the particle count parameter, you can gate on images with 1, 2, or more particles in an image. The imaging parameters improve analysis in the downstream gates. If we look at the scatter plot and images in the singlets gate, manual gating would have excluded the target cells, and you can see this on the top right. On the other hand, if we looked at the scatter plot of what we would consider singlets, then we would have included off-target cells. And you can see this on the bottom right. This type of gating is subjective, thereby reducing reproducibility and increasing analysis time. We can help to improve it. You can use label-free morphology parameters to separate populations based on differences in particle size, or cell texture, uniformity, or complexity. This gating scheme suggests that we can distinguish between beads and cells. Beads appear very dark in the images and have less complexity, and they are more uniform compared to cells. Entropy intensity refers to the complexity in light and dark spots with the event, and these are interpreted by the model as different populations. Events that are high for average normalized intensity tend to have more bright or white spots. You can see how healthy cells have higher average normalized intensity compared to beads and unhealthy cells, which may have more dark spots. You are viewing how the model has created a hierarchy within the sample using thousands of images. This analysis is label-free and does not require the use of fluorescence or scatter parameters. Using the same experiment, you can customize your analysis based on morphology and gain more resolution compared to traditional flow cytometry. Because the CytPix is fast and the sample volume is up to 1 milliliter, this is actually pulled into the instrument at one time. And because of this, the beads will not have time to settle to the bottom of the tube. This analysis within the tube can be completed in seconds. The intensity features can be plotted against each other so you can see how the populations are separated based on morphology. This time it's pixelation. In this example, you can see differences between populations that have only beads, only cells, and cells with beads. You can exclude debris upstream in the analysis and our quality control gates, which are not shown here. Imaging and morphology parameters can be used to accurately track the purity and health of your cells post-sort. In this example, we stained induced pluripotent stem cells with Tri-160, a marker of pluripotency, and then also with CytPix red as a marker for viability. We then used the Bigfoot spectral sorter in single cell sorting mode and compared the Tri-160 positive cells and CytPix negative populations. And you'll see there in the bottom plot, we're really just looking for that particular population. And so we compared this population pre and post sort using the CytPix. And using the particle count parameter, we can see that we have an increase in singlets post-sort. You can see that on the bottom left, compared to the pre-sort sample in the top left. We can use the shape feature, eccentricity, to discern between healthy and stressed cells. The more eccentric shaped cells has this shriveled morphology, and we were able to exclude many of these in the final sort, thereby increasing the number of more round cells, which appear to be healthy. And you can see that the increase in percentage of healthy round cells in the bottom plot here, and how these look and compare to the more eccentric, less healthy cells in the top right. If you want to confirm the occurrence of rare populations, this is also possible. And in some cases, this can suggest physiologically relevant events. CytPix image analysis can also capture and quantify the cell interactions, such as this shown in the CAR-T cytotoxicity assay. Ramos cells and CAR T cells in quadrant 1 and quadrant 4, respectively, are stained for tracking purposes. The double positive stained cells in quadrant 2 can be visualized to suggest a possible immune synapse. Without the image data and morphology parameters, the analysis of the cytotoxicity assay would stop here on a traditional flow cytometer. You can see these plots on the left. We can add depth to this analysis by quantifying which events comprise of true interactions versus coincident events. Again, the shape features and intensity features in this example, circularity and skewedness, are used to show which events have double stain positive cells that are attached versus those that are unattached. Our no-wash, no-lyse assay successfully eliminates erythrocytes from CD45 positive leukocytes in analysis of whole blood samples. Often, it is difficult to identify populations in complex samples and exclude contaminating events. Unwanted events, such as doublets and aggregates, may also pick up some of the stain. We can use the particle count parameter to discern singlets and gain better agreement with surface staining. Using a simple singlet gate, particle count is equal to 1, as I showed before, we can exclude unwanted events. The parameters can inform and improve our gating strategy. Because of the complexity of this sample, we're combining unlysed red blood cells and leukocytes. We still won't have a perfect gate, but we can see that the morphology parameters will improve the accuracy of our downstream analysis. So you can see here that we can actually get more CD45 positive leukocytes using the image morphology analysis. The Attune family of flow cytometers also has robotic automation and accessories that enable long hours of continuous runtime. There is a CytPix Max auto sampler that can utilize 96 and 384 well plates. The orbiter can move plates to and from the storage towers. Combining label-free morphology analysis with speed and high throughput ability on the CytPix can help improve the efficiency of your lab. You can visualize and confirm that rare events are cells, standardize and reduce user variability and gating, discriminate and achieve better resolution in populations with confidence to help improve data quality. And finally, deepen biological insights while witnessing physiologically relevant events that are indicative of cell behaviors. Thank you.
Thank you, Nicole and Jasmine, for your presentations. We'll now start with the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. You can put those into the Q&A box on the screen. We'll answer as many of your questions as we have time for. So let's get started. The first question that has come in is, how does integrating marker expression data into morphology improve predictive accuracy over morphology-based analysis alone?
I'll take that one. Yeah, good question. So combining immunophenotyping with morphology analysis provides a more comprehensive view of donor material by integrating functional marker expression with the structural attributes of the cell. Marker expression offers biological insights into cell identity, activation, and function, while morphology captures physical and structural data. So when you combine them together, they can enhance the resolution of the predictive models by correlating physical traits with functional states and distinguishing between similar morphological profiles with different roles. As a result, we can gain deeper insights into donor subpopulations that may perform better or worse through the workflows.
All right, thank you. So the next one we've got is how can predictive morphology-based models alleviate bottlenecks in the manufacturing process?
I can take that one. Predictive morphology-based models offer potential to streamline manufacturing by enabling early screening of donor material at day 0, identifying suboptimal inputs, and conserving resources. They can guide smarter allocation of reagents and manufacturing capacity by linking things like morphology parameters to expansion potential, thus facilitating reliable dose reductions. These models might also support tailoring of protocols to donor-specific characteristics, such as adjusting activation or culture conditions for heterogeneous populations. And by standardizing processes and correlating morphology with dose efficacy, these approaches can reduce variability, prevent batch failures, and ultimately enhance efficiency, scalability, and cost effectiveness in manufacturing.
All right. How could incorporating additional markers, exhaustion, activation, maturity, improve workflow outcomes?
That's a good question. So, incorporating more markers will enrich cell profile data and provide deeper functional insights. For instance, exhaustion markers might identify cells less likely to sustain function, while maturity markers could indicate readiness and education of donor cells, or whether a certain part of the process pushes the cells towards a certain maturity subtype. Combining morphology and these functional markers can help refine donor selection, identify target cell population for sorting, and enable better workflow optimizations. It also really does improve the predictive power of the models, which enhances product quality and ensures greater consistency through the workflow.
Okay. Another one that we've got here is how could integrating morphology analysis into the donor selection stage influence long-term outcomes for cell therapy products in the autologous and allogenic space? I apologize for that.
No worries. I can also take that one. It's a really good question. So, for allogeneic therapies, where one donor can produce multiple doses, morphology analysis can identify high quality donor material by assessing parameters such as circularity, texture, and size, which may predict expansion potential and therapeutic efficacy. By enabling the selection of more consistent donor material, manufacturers can better achieve predictable scalability and batch-to-batch consistency. Over time, correlating these morphological metrics with therapeutic outcomes can enable more adaptive workflows and enhance cost efficiency. For autologous therapies, where the starting material comes from the diseased patient, morphology analysis can be used to really guide tailored manufacturing strategies and dynamically monitor these vulnerable cells through manufacturing. Early assessment can give foresight into potential cell vulnerabilities and opportunities to adjust the process ahead of time. For instance, morphology combined with marker data could inform potential rescue strategies or allow for selective enrichment of viable cell populations. Additionally, the morphology data can also help set realistic expectations for therapeutic efficacy, especially for these compromised cells, and inform researchers, patients, and clinicians on potential adjunct therapy decisions to enhance success for the patient.
All right. The next one here is what is the sample throughput for CytPix?
Hi, I can take that one. So the CytPix It can use tubes or plates. So it can use tubes with volumes up to 4 milliliters. So from very small volumes up to those larger volumes compared to other flow cytometers. And with the CytPix auto sampler, you can actually analyze samples in 96 well as well as 384 well plates. And the 96 well plates in high boost mode can be analyzed in as little as 22 minutes.
Okay. We've got time for one or two more here. How fast does image processing occur?
Oh, that's a pretty simple one. So really, it's also dependent upon the size of the image data. So the image processing itself can analyze the images up to 1,000 images per second. And that is based on a 248 by 248 pixel size.
Okay. And we'll end with this one, but like I mentioned before, any questions we don't get to today, we'll follow up with afterwards. Or if you're sending a question through during the on-demand period, those will also be followed up with. So we'll end with how many different markers can be measured by Attune at once?
The CytPix is able to measure up to 14 different fluorescent colors. And as you can see, as morphology emerges, there's another set of markers where you can learn more about the cellular responses. So now we've got two data sets. We've got morphology as well as the traditional flow parameters, which are fluorescent and then also scatter. The sky's the limit.
All right, excellent. So I would like to thank you again to Nicole and Jasmine for your time today and your important research. I'd also like to thank Labroots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us and their questions. Questions we did not have time for today, and for those submitted during the on-demand period, we will follow up with those via the contact information you provided during the time of registration. This webcast, like I mentioned, can be viewed on demand, and Labroots will alert you via email when it's available for replay. We do encourage you to share that email with any of your colleagues who may have missed today's live event. And until next time, thank you very much and good.